Today I am going to tell you a story of Ruhi, a new researcher in a molecular biology lab. Ruhi is a dynamic, meticulous planner and a very sincere student. She wants to clone a gene, and as a first step, she isolated the genomic DNA. To check whether her DNA is perfectly alright or not, she used a agarose gel electrophoresis, a standard method to check the DNA consistency. When she ran the gel, she could able to confirm that the protocol she followed for the isolation of the genomic DNA was perfectly alright. With this success, she went ahead and she set up a PCR, a polymerase chain reaction. Now, this PCR reaction is based on a standard predefined protocol which is existing in her lab. Essentially, she used the buffer, the DNTPs, the primers, the template DNA, the tag DNA polymerase in a reaction volume of 20 microliter. This was a whole day lot of work for her, and next day she came. Just to tell you in briefly, for her PCR she used the initial denaturation step, a denaturation, a primer annealing and primer extension and a final extension step. I will be talking about this in a minute. Now let's focus here about the Ruhi's PCR results. While she ran a gel, the same agarose gel electrophoresis, and to see the amplified product, she was very excited before running the gel. When she ran the gel, she observed something this. That is, that she could able to see the markers which indicates that her gel and the ethidium bromide used for her gel, the gel casting and everything seems fine. But the lanes which she used for adding her PCR samples, it is blank. There is no amplified product. Ruhi was very disappointed and she was having no clue that why this happened. If you are a researcher in an early career and you are working in a molecular biology lab, so I welcome all of you to Mannan's e-learning program. In this series of the lecture, which is a technical lecture, I am going to talk about the PCR troubleshoots. If you do not know me, my name is Mannan. I am a PhD in molecular biology. Before I talk about what are the troubleshoots or the problems which you face in PCR and how you can able to solve, let's briefly see about the PCR or a polymerase chain reaction. If you see here, the polymerase chain reaction, you basically use the DNA, and in case of Ruhi's, if we take an example, then it is a genomic DNA. Then you need the DNA primers. As you can see here, one is a forward primer and one is your reverse primer. Besides this, you need the buffer, the optimum annealing and denaturation conditions. You require the DNTPs, that is deoxynucleotide triphosphates, that is DATP, DCTP, DTTP and DGTP. Okay, this all is required, and it essentially consists of a initial denaturation step where your DNA is being separated, means that hydrogen bonds is break. Then the primers comes and bind on the basis of the complementary over the sequences. These primers then are been amplified by the enzyme, that is DNA polymerase, and this DNA polymerase could be a tag DNA polymerase, it could be a Vent DNA polymerase, turbo DNA polymerase, or any DNA polymerase depending upon the user's use. After this, your PCR cycle starts running. So essentially there is a initial denaturation step where you basically separates the DNA. Then these steps, that is the denaturation, annealing and extension or the elongation steps, is repeated from 25 to 35 cycles, and then your PCR is been done. PCR is one of indispensable technique, and this technique is, you can say, invented by Kary Mullis. And the Kary Mullis, who is no longer with us, but his invention with us, any molecular biology lab is cannot, or any molecular biology experiment cannot be performed without polymerase chain reaction or the PCR. Now we will be, as I said, this whole lecture is about the problems which is associated with the PCR. Now if you see, there is a gel picture number 1 which has been shown here. You see a PCR product. The left and right hand side is the markers which has been shown to you. Now this PCR product seems is fine because you all get the amplification. However, if you look carefully, that is the lane number 1, the lane number 3, apart from this marker, and this lane number 5, you see that the yield of the PCR product is less. If you compare this with the lane number 7, you also see there is a small smiling effect over here. The products with this is also more. Now for a routine PCR amplification step, this gel is perfectly fine. However, if you want to do a downstream processing in the sense, like if you want to cut this DNA, and say if your product DNA is very less and you want to do a gel extraction, means you have to cut out the exact size of band, and then you have to take out this DNA from the agarose gel. There will be a loss of 20 to 30%. There will be loss, and in some cases it might go to even 50%. Now if your product is less or your yield of product is less, then you can imagine that there could be a problem. The other problems could be associated with a non-specific amplification. If you see here in the gel number 2, you are also seeing a band which is the lower size of the desired PCR band. This is the desired PCR size band, which should be like 130 base pair, but you're also getting a small fragment of bands. Or there is also possibility you can see that these higher molecular weights bands is there. So there could be several problems which could in the PCR while for performing a PCR. Anyone who is doing a PCR, they might face this type of a problem. In fact, I also faced a problem during my PhD days in PCR. In one way, it is good for Ruhi or someone who is working on a PCR, because if she would have got the bands, then she would have gone to her next step, and she may not be watching this video, or you might not be interested in watching this video. But if you are watching this video, it means that you are having some kind of problems in PCR, or you want to learn the concepts that what could be the, or you can prepare ahead that what could be the problem and how to solve this problem. So the problems in PCR could be the low yield, like I showed to you. There could be smear, means there could be a smear, means whole smear kind of a thing can also happen. Or there could be a non-specific amplification, or your desired size of band is not occurring. Now, having said this thing, I will try to focus here only one of the problem of the PCR, that is no PCR band, because that is the major problem, that if someone is not having a PCR amplification. There are as you can see in this picture, and this picture is something is from my own research. When in my early days, when I was struggled with the PCR amplification, I got the PCR band in this gel. You can see this here is the yield is low, and in this gel there is no amplification. So everybody faced this problem. When I invert this picture, and you can always do by inverting this picture, and I can tell you maybe in another lecture how you can do, but essentially what I did, that the image contrast and inverted this image in a paint 3D software. You can use the other software as well. So the ultimate bottom line is that, and which we are trying to discuss, that what happens if there is a problem that you are not getting any kind of yield in your PCR. Several companies, because whenever we do a PCR, we usually buy the PCR reagents in terms of the primers, in terms of DNTPs, in terms of enzymes from the companies, and these companies also gives you the troubleshoots for them. Like for example, let me skip this. This is one of the URL I will also share in the link, so that you can click and follow this. This is from the Merck, from Sigma Aldrich, because Sigma Aldrich has now been merged with Merck, the Thermo Fisher one, and the NEB one, that is New England Biolabs. You might find, depending upon which vendor you are using, and I'm here not to promote any vendor as such. I'm just telling you there are certain companies, and you can go into the links and find out what kind of exact problems is there. For the short of time, and one of the major problem, I'm just focusing only one of the problem, that is your no amplification occurred. So let's see that what could be the problem, why you are getting a no yield, or why you are not getting any amplification. The possible reasons could be that it could be an incorrect annealing temperature. Means if the suggested temperature, which is say let's say 60 degree, and if you are doing your PCR maybe at 65 degrees Celsius, or maybe a too low temperature like this 50 degrees Celsius, then there could be a problem. The other major problem which can occur due to a poor primer design, means the designing of the primer is one of the important criteria even designing a PCR, or even designing any experiments one has to design. So that could be a problem. Maybe there could be a hairpin loops, that is a possibility is there. There could be internal complementarity bases could be there, means they can able to form a primer dimers, and then there could be no PCR amplification. That could be a problem. This is one of the problems we are trying to see what could be the problems. Now to solve this problem, one can use a gradient temperature, and for running this gradient temperature, one can use a gradient PCR or a thermal cycler machine. If you do not have, there is nothing to be worried. You can design, you can set up your reaction for maybe 1 or 2, let's say your ideal temperature of your TM or for primer annealing is 60 degrees, then you can set up your PCR reaction at 65 degrees Celsius and say 55 degrees Celsius, and see where you are getting your PCR bands or the desired band. For primer designing, there are several softwares which are available. You can use Gene Runner, like this is a software we use often in our laboratories. You can also use Vector NTI, though this is commercially is not available, but you can use. You can also use CloneTech PCR software for this. You can also use the NEB PCR designing tool as well. I will there is other softwares like Eurofins is there, and I will share that link with you. So verify that there is no complementarity which has been there, means you make sure that there is a non-complementarity both internally and to each other. When I say internally, means say let's say this is your say primer, let's say I'm arbitrary writing, means they should not able to form a means like say for example this one, the CGG can able to bind with this, so that is called as a internal looping. That should not be there. The other solution to this, one can increase the length of the primer. Besides this, there could be other problems. The problems could be like your poor primer specificity, means that the primers can go and bind to some other place, or the primer has been degraded because of the freeze thaw methods. That's why it is always advisable that whenever a primer which comes into a lab, when the original stock, like for example in our lab we used to make a stock of a primer 100 micromolar solution in TE buffer, that is tris EDTA buffer, and then we make a working stock of this as a 10 micromolar. We aliquates this into several 50 microliters or in fact 100 microliters vials stored at in minus 20 degrees Celsius or minus 80 degrees Celsius, and then use one vial so that to avoid the primer degradation. Because you might be thinking your concentration of primer is fine, but because of the freeze thaw there could be slight degradation, and then the concentration of primer could be less. Though its chances is less, but that could be also possibility, and that could be the reason. And to solve this problem, one can verify the oligos complementary with the target sequence. You can use the MCBI software tools for this, and you can see like by putting your whatever the gene sequence in a FASTA format, and then you can search whether your primers can bind with that or not, and you can use a optimal concentration of primers to solve that. You can also search for a specific literature information if someone else has worked upon. The other possibility which can also happen sometimes, that is you forgot to add the missing components. Now usually or the practice which I used to follow, like say suppose if I have a buffer component, if I have a primer component, if I have let's say DNTP component, if I have a water component, if I have an enzyme component, the practice which I follow is that whenever I set up a PCR reaction, I follow a sequential order, that is the enzyme to be added at the last, and then and check mark that yes, I added buffer, I added the primer, I added the DNTP, I added the water, and I added the template, okay, because that is what is been required, and then I added the enzyme. Check my reaction condition in the PCR machine is perfectly all right or not. So one can repeat the step, but the repetition merely is not solve the purpose unless you follow this checkerboard method. You can look into the magnesium concentration which is present in your buffer. In some laboratories they add because this buffer is with the magnesium or they are without the magnesium, so make sure the buffer is having the magnesium. Usually it's range from 1 to 2 millimolar. But if it is not added, make sure it has been added into the buffer. Or one can optimize the concentration of the magnesium as well, because this magnesium as a 20x or 10x vials is very easily available in a 25 millimolar concentration. The companies and the vendors properly they sell these things. So you have to check this, you have to make this checkerboard method. Don't use your intuition. That better to use a pen and pencil in your lab notebook and try to do this thing. The chances of error will be very less. The other problem which can occur is the poor template quality, and I would consider this is one of the prime reason if you are meticulous, if you are a sincere reason, and to check the quality of your template or the genomic DNA, it is advisable that one should use a nano drop machine by taking the 260, 280 and as well as 230 measurements. If a nano drop instrument is not available, then one can also use a spectrophotometer to check that as well. okay, and as well as check by running an agarose gel and see that there is no inbuilt protein or the protein contamination is there. If that is so, then it is advisable that you use a ethanol precipitation step to clean your genomic DNA precipitation step, or you can also follow the kit for isolation of genomic DNA. The advantage of using a kit is that you can also get rid of these inhibitors. There is a possibility that there is a insufficient cycle, but among all of this, I still consider that poor quality of the genomic DNA based on my experience is the culprit. If you could able to solve this, then this no amplification can be turned into yes amplification, means you will get the PCR product, and we are trying to solve this in this lecture. So these are some of the tips based on, and all these tips is based on the NEB websites. You can visit them, that you analyze the DNA, you check out the ratio, you start with the purify, you decrease the sample, means if you are using say a 50 nanogram of DNA, maybe because of the impurities which is present or the inhibitories which is present, your PCR is not occurring, then you can reduce it to a 5 nanogram or in fact 0.5 nanogram of DNA as well. This has been shown that if you dilute your template DNA by 10x or by 100x, the chances of success also increases. You can also decrease the volume size, means the when you are isolating the genomic DNA, the genomic DNA yield should be perfectly all right, means you if you take the concentration of the DNA, it should not be like for a 50 nanogram reaction which you are using, and you are using say 10 microliter in a 20 microliter reaction of genomic DNA. That is not advisable. Maybe you use 1 or 2 microliter of your genomic DNA in terms of volume. If you rerun the cycle with this precaution, there is a chances that you might get a success, because ultimately for a PCR you need a single molecule. There could be other possibilities which has been mentioned here, that it could be a incorrect thermal programming. This happens sometimes with us, that you run the PCR, this is a predefined you can say the protocol which has been there, but somehow somebody else has changed that. So it is always advisable that when you put your samples in the thermal cycler, wait for 2 minutes or 5 minutes and see that it is working fine. Most of these thermal cyclers now which is coming, they are coming with a LCD display, that whatever the reactions which is occurring can be easily seen outside. They can show you what is the cycle is running. Look for the block temperature. There should be no contamination from the reaction tubes, that is you use gloves hands, and preferably a powder free gloves for setting up the PCR, because sometimes the DNAs might come from your hand. There could be a possibility that could be a complex template, the GC content of your product, the genomic DNA could be more, then you have to probably add certain additives, like glycogen is one of the additive which has been used, DMSO which has been used for this, and then you might get success with that. Besides this and the requirement of your purpose, you might also think of using a high fidelity polymerase, because tag DNA polymerase, like in case of Ruhi, is not advisable for cloning purpose. Either you use a very high fidelity polymerase, you can use a GC enhancer polymerase if you have a complex template, or if you have a product size is of long, that is more than 3 kb or so, then you might think about a turbo polymerases which has been there. You can also avoid this by hot start polymerase PCR technique as well. Dear friends, I would like to tell that problems in laboratories might occur, but what is more important is your attitude, your thinking. And that's why it is always said that patience is one of the key. One has to be patient. Whatever the problems one is occurring, you should be patient. And once you is having a patience, then you second thing which comes is your perseverance, that you don't give up the things. As Julie Andrews says, perseverance is falling 19 times and succeeding the 20th times. So maybe you use this as a stepping stones to learn new things, you explore new things. Don't give up, and you will be succeed, or you will get success in your endeavor. So whether you are a Ruhi, you are a Richa, or you are a Rohan, Rihan, Rohit, whosoever is you are, if you are listening to this lecture, I wish all the best for your experiments, and I want that you all should become success in your career by learning these technical skills. With this, thank you very much. If you like my video, please subscribe to my channel. Thank you.